Welcome friends, myself Shoptaporni from BioLive. Hope all of you are doing very well. So today I am going to teach you about Biuret Test or Piotrowski's test. What is this test? It is a quantitative and qualitative test for protein estimation. That means if someone gives you an unknown solution and asks if there is protein or not, you can do this test. And then if they ask how much amount of protein is there, you can also estimate that, okay, by using a colorimeter. And if there is protein in the range of 5 to 150 milligram per ml, then this test could answer the amount of protein present in your solution. Okay. Now, histidine is the only amino acid that takes part in this reaction. Remember that single amino acid or dipeptides never take part in this reaction. At least 3 amino acids are needed for the positive test. So, how does this reaction gives the positive test? Now only remember, lone pair of electrons of nitrogen atom of the peptide bonds actually take part in this reaction. How, we will see later. Now, why this reaction is named Biuret test? Biuret, bi means 2. See, if there are excess urea, this is the formula of urea, CONH2 twice. Okay, you can see there are 2 molecules of urea. Now, if there is excess urea and you heat them at 180 degrees Celsius, then they will form the biuret complex. And here see, this is the biuret complex and they will relieve the ammonia. In this biuret complex, this part CONH is exactly same to the peptide bond of our protein molecule. Okay, that is why the peptide bond and biuret complex gives the similar result when they react with the biuret reagent. Okay, that is why this reaction is also known as Biuret test. Though there is no presence of urea in this reaction, but the similar kind of peptide bond is present both in protein and biuret complex. That is why this reaction is called Biuret test. Now, let us see what are the components of biuret reagents. See, these are the components of biuret reagents. What are those? 1% copper sulfate solution. This is the cupric ion. Okay, CuSO4 5H2O, which is of light blue color. Then 5% NaOH solution, which will make the medium alkaline, and copper sulfate solution will provide Cu2 plus or cupric ion. Next, potassium sodium tartrate, which is also known as Rochelis salt. It is a chelating agent. It will chelate and stabilize the Cu2 plus ion with the peptide bond. Okay. Now, you need to add 1.5 gram of copper sulfate solution, and then 6 gram of Rochelis salt in 500 ml of distilled water. After that, add 375 ml of NaOH in that solution, and at last make up the solution up to 1 liter by adding distilled water. Okay. Now, sometimes potassium iodide can also be added to this solution to inhibit the reduction of cupric ion to Cu plus, but this is an optional step. Okay. Now, how does these components help in the reaction? Let's see. See, this is the BSA solution. BSA is nothing but the bovine serum albumin, which is a standard protein solution. Take 1 to 2 ml of BSA solution and take the biuret reagents, which is also of 1 to 2 ml. And when they will react, they will show a purple or violet color formation. Why this purple or violet color forms? Because of the formation of copper protein complex or biuret complex, which is also known as coordination complex. This copper protein complex actually absorbs visible light at 540 nanometer and gives the purple or violet coloration. Okay. Now see how this reaction gives the coloration. What is the basic or chemical basis of this reaction? Biuret reagents react with the peptide bonds present in the protein solution in the alkaline medium. The alkalinity is given by NaOH, okay, and forms a purple complex. Okay, here the cupric ion of copper sulfate forms a coordination complex with lone pairs of electrons of 4 nitrogen atoms of peptide bonds, and the tartrate of Rochelli salt or the sodium potassium tartrate acts as a chelating agent in this reaction. See, this is the protein. Okay, this is the N-terminal end, this is the C-terminal end, and this is the peptide bond, CONH. This is the peptide bond. Okay, as this is the protein, we have given it a N number. Okay, and when you will add the Biuret reagent, that means the copper sulfate and NaOH, the alkaline medium, it will form a in presence of what? In presence of Rochelli salt, the tartrate. Okay, sodium potassium tartrate. The tartrate will help to chelate the Cu2 plus, the cupric ion, with the lone pair of electrons of nitrogen atom of the peptide bond present in the protein. Okay, and thus Cu2 plus will chelate the peptide bonds, the nitrogen, the lone pair of nitrogen of the peptide bonds. Okay, and it will form a complex known as copper protein complex or coordination complex, which will absorb the light at 540 nanometer and will give purple coloration or violet coloration. This complex is known as purple coordination complex or Biuret complex or copper protein complex. Now, the intensity of this purple color or the violet color will depend on the number of peptide bonds present in the protein solution. As the number of peptide bonds will increase, the intensity of the color will also increase. As we know, only histidine reacts in this takes part in this reaction. So, more number of histidine means more intensity of the color. Okay. Now, last but not the least, in the positive test, obviously you will get a purple or violet color due to the formation of copper protein complex, this complex. But in a negative test where no protein is present, obviously this complex will not form. So you will only get the light blue color of the copper sulfate reagent itself. Okay, so in positive test you will get purple or violet color, but in negative test you will only get the light blue color of the copper sulfate in absence of protein. Okay. So this is all about Biuret test. I hope this video will help you. So thanks for watching, and don't forget to like, share and subscribe. Thanks for watching and happy learning.